Hi, welcome to Virus Appreciation, or maybe this is weird sci-fi art appreciation class. Yeah, check out our mascot today. A cool little virus that I think is mutating. I think that's what the electric blue thing going on in there. It kind of looks like a coronavirus that picked up a protein called a hemagglutinin esterase, which is what I think the little blue ones are. But you know what? Yeah, art doesn't have to have reason and science behind it. Science does. However, what we're talking about today is kind of out there. So, we're talking about the origin of viruses. And it's an area where there isn't necessarily a right or wrong answer, and there may be multiple answers. But because we have very different kinds of viruses. So remember back to the beginning, we have all these different realms of viruses. I think there are 7 at the moment, but if you're watching this in the future, maybe there are 25. And those realms of viruses have nothing in common with the other realms. So they appear to have independently evolved from something. That something may have been another virus. It may have been something that was less than a virus that sort of broke out. It may have been a cell that just jettisoned all of its worldly cares and most of its genes and adopted a viral lifestyle. And it's possible that all these things are true for different groups. So, different realms of viruses not related to each other, just clustered together in this course so that we can talk about all of them, because where else are you going to talk about stuff like viroids? Yeah. Plant biology class, for one thing, plant pathology. But I don't know. I don't know if you're going to be there. So let's go. Let's go. Let's get into it. The 3 big hypotheses are that viruses are basically less than cells, that these things predate cells. That's the virus early hypothesis. Because a virus has usually a replicase, like something that can copy the virus. Most often, this is going to be a polymerase. Occasionally, you get weird viruses like HPV that don't even bother to carry a polymerase anymore, but they've evolved enough little tricks that they can get themselves recognized and copied by the host cell polymerase. It imposes a big sort of lifestyle restriction. They can only go into cells. They have to go in a certain way. Once they're in these certain types of cells that are actively dividing, they have to wait for active division to happen. So, yeah, papillomaviruses work in certain contexts pretty well, and in other contexts, not at all. Yeah. But they work well enough that they exist in the world. The regression hypothesis is a neat one. So there are some very big DNA viruses. One group are called the Phycodna viridae, Phycodna, I don't know, viralis maybe. And these are large double-stranded DNA viruses that infect usually algae, but you can call algae like degenerate single-celled plants or primitive plants. So there may someday be one that infects a vascular plant. And also things like mimivirus. That's the internet's favorite giant virus, or some people call them gyruses. And these are things with hundreds to thousands of genes which have genomes that are bigger than some bacterial genomes and are nevertheless viruses because they don't encode enough equipment to be able to sort of self-sustain, to be able to do it on their own. Now, a thing that we'll get into in a minute, there are also bacteria that we know have lost some of these components. Things like mycoplasma is one kind. Or rickettsias or chlamydia, yeah, the sexually transmitted disease, that's a different kind. These are bacteria that have lost just enough genes that they can no longer fend for themselves unless they are inside the cytoplasm of a cell. And doesn't that sound a little bit like a virus? Yeah, I think so. So there are, larger viruses, things like pox viruses that could conceivably have come about this way, or else they are excellent collectors. They've been out grabbing little genes from everywhere and building up a collection. But in any case, all these things exist right now and in many different forms, and we are probably hundreds of millions, probably actually billions of years from the real origin of most of these virus groups, as far as we can tell. The study of fossil viruses is not something that has been done particularly well at this point, just because it is really hard to get something like a virus from a fossil, or even to show that there were viruses there. They have viruses from thousands of years ago, some of which they've managed to boot back up. Viruses from hundreds of thousands of years ago, where they can show that, hey, it's here, it looks like a kind of virus that we know, and it no longer boots up, possibly because it's damaged. Just that long in the UV radiation from the sun will not do good things for your RNA or DNA genome. And viruses, at least the ones I'm aware of, do not have RNA or DNA repair mechanisms built in, because they are like, you only live once. They're little YOLO creatures. They live fast. They die young. That's just the virus way. Even the gigantic viruses are some version of that. They're all sort of skating on the edge of destruction. Yeah. Anyway, the third one is that viruses are actually escaped genes. And interestingly, you can find in many different eukaryotic genomes what looks like an ancient, it's kind of like a retrovirus. So it'll have something like a reverse transcriptase that's related to the coronavirus polymerase. And right next to it will be something like a helicase that is related to the coronavirus helicase. Now, are these the ones that somehow came back out of the genome and picked up a totally different set of structural proteins, like the things on the outside of the exploding neon blue ball up here? Or did they have a totally other origin? Hard to say, actually, possibly impossible to say, but I don't know. Somebody may come up with a clever experiment at some point. The other problem here is that you're probably thinking, well, fossil DNA. Yeah, gotcha, yeah. Even under optimal conditions, like in the darkest, coldest cave and proper, frozen basically, we think the longest that DNA can stay intact enough that you can even read the little tiny fragments it breaks into is somewhere around 6 million years. I think the oldest DNA that has been credibly read is a little older than 1 million years. Things like proteins last a little longer, so you might be able to detect that. But viruses change so fast that their proteins change pretty fast. And so I don't know that you'll be able to get any good answers to anything in this lecture. The point of this lecture is not to actually answer the question, where do viruses come from? The point of this lecture is to look at a bunch of different viruses and how they work and kind of get a scope for what's out there. Because if you can imagine a thing, a virus can do the thing. And viruses also do some things that you probably didn't imagine. I certainly didn't imagine. So yeah, let's get into this. Let's get into the origin of viruses. Here we go. Okay, so here are some of those obligate intracellular parasite bacteria. And each of these are names of, I think they're all genera. So this is a genus. Coxielas cause Q fever. Chlamydia is chlamydia, that you know and love and hopefully don't have. But one of the things it causes is basically blindness that you get as a sexually transmitted infection. That's kind of one of the end games of chlamydia. It's terrible, yeah. And rickettsia. Sometimes bacteria like this are grouped in the Rickettsia or whatever. But these are some of the little stats here. And these are all bacteria that can't survive if you try to grow them in like chicken soup or, bacterial growth medium. There's no way to keep them alive. They require some things that they can only find inside of eukaryotic cells. And I think all of these are exclusively vertebrate animals that they infect, as far as I'm aware. Actually, no. Rickettsias are much broader. Those infect ticks for sure. Actually, yeah, and so Coxiella is also zoonotic. So never mind. Yeah, they infect a lot of stuff because they're just bacteria. And they are related to bacteria. You can get out the genes that they have, and you can put them on bacterial trees because they are the same gene and they come from the same ancestor as the version of the gene you would find in other bacteria. The genome size is worth paying attention to. So 1 to 2 million bases, that's what we're looking at here. And yeah, that's pretty reasonable. There are some bacteria that go up to like 6 million, and there are exotic ones that are much bigger than that, I think. But this is about right. So when you're thinking about bacterial genomes, yeah, 1 to 2 millions. That's the ballpark. All right. Neat thing about rickettsia is that there are rickettsia bacteria that are actually required by their tick hosts. So they don't cause disease, at least as far as we understand, but they do make it possible for the ticks to actually live and breed. So that's kind of cool. Yeah. We think maybe mitochondria came from this sort of origin. So when studying all of these things, these origins, what you need to do first is identify the genes that are the same gene. And there are statistical ways of doing this. You can say that these are close enough that they are closer to each other than they are to anything else. And there are different mathematics that allow you to do this, but what you're looking for are things called orthologous genes, or conserved orthologous genes. That's this COG you see right there. Yeah. And so these are genes that are, basically either stolen from an organism, but it's the same gene, or descended, from a common ancestor, but it's the same gene. And they're probably going to end up doing the same job, and they're going to end up having the same shape, the same fold. And so, by first compiling a list of these, then you know which things you can align in your bioinformatics class. And then once you have your alignment, you can make a family tree for each of those genes or for all the genes together, whatever. And that's how you explore, like, where did this gene come from? Where did this organism, what is this organism related to? That sort of stuff. So like I said, if you look at a simple pathway, like, remember from Biology 1, remember glycolysis? So these are the enzymes. Yeah, glucose 6-phosphate goes to fructose 6-phosphate. Oh my gosh. This is how eukaryotic cells, actually, and prokaryotes, I think, use glucose to generate ATP, basically. And these are the enzymes that are able to do this. And so for most of these steps, for some of these steps, there is only one enzyme. So you need a phosphoglycerate kinase is, I believe, what the PGK is. And all the phosphoglycerate kinases in all the animals that exist, each of the letters is a group of different genomes. Those are all related to the same thing. Same thing for down here. This is an enolase, and all of those share a common origin. For some of the other things, we have replacement over time. So you'll either have an FBA type thing that's going to act on fructose 1,6-bisphosphate, or you'll have a DHNA type. But those are the two types, and it's just like this is probably the ancestral one, this is probably one that replaced it and did the job as well in some other group. So anyway, these would be considered orthologous genes like this one and this one. The gold ones essentially are the original ones that are found in most cases. All right. So if you want to look, if you're just curious, what are the orthologous genes that you would find in living things like bacteria and archaea and all the eukaryotes? Yeah, a banana and an E. coli and you. What do all these things have in common? And so there's a list. There are at least 63. This is the most conservative list that I have seen, which says that, there are at least 63 genes that you find in every branch of life. Because if you think about it, every living thing, as far as we know, has DNA as its genome, has RNA, messenger RNA that it makes, translates it with ribosomes, and has some means of acquiring energy and making it in the world. Now, the acquiring energy and making it in the world, things like metabolism, those can be done a lot of different ways, and so you'll see a lot of specialization there. But the genes that actually encode the important stuff, like copying your DNA or processing proteins, stuff like that, even making some of the components for those tend to be real conserved. So like of those 63, almost half of the conserved orthologous genes are part of the ribosome. And the ribosome, you remember, is the thing that translates messenger RNA to make all the proteins that we need, that everything needs. And the ribosome is conserved. And also the ribosomal RNAs are also conserved. Yeah, definitely. The things that make the tRNAs tend to be pretty conserved. Those are conservative little proteins, and you'll have multiple copies of most of them that do slightly different versions or are turned on at slightly different times and turned off. But yeah, it's we're essentially doing the same process to achieve the same goals as chlamydia. Yeah, oh boy. Yeah, is that a group you want to be in? Probably not. I don't know. And so on. So yeah, basically, the ubiquitous genes are involved in really basic processes that are here. So now, if we're applying this to viruses, what are our ubiquitous genes? And the answer is that, well, there aren't any that cover all the viruses. So there's no one gene that you can find in every kind of virus. There are several different realms of viruses and virus-like things, and in each of those realms there will usually be one or more genes that are conserved throughout the realm. Or even if you find one weirdo that's missing the one gene that it should have, if everything else is close enough to other members of the group, then you can say, all right, we will include this in the same group. Yeah. So if you're thinking viruses, what's our conserved thing? Maybe it was the first question on your second exam. Maybe it's one of the things we've been hammering on since day one. What about the polymerase? And yeah, not all viruses have one. And not all things that we classify as viruses have one. But most of them sure do. Yeah. And when they do, they all kind of look the same and work the same. So do you remember your polymerase structure? You had your palm and your fingers domain. The fingers are alpha helices. Look at these little chubby little alpha helices. And you got your thumb domain, which is also usually alpha helices. And the palm, yeah, look at those little things in there. That looks like a beta sheet to me. It is a beta sheet. It's a beta sheet that usually has 4 strands in it. And yeah, that is where the active site of the polymerase is. Turns out, if you look at polymerases as a whole, so here we have reverse transcriptases, RNA-dependent RNA polymerases, and different kinds of DNA polymerases, for example. Not even DNA polymerases, but different kinds of RNA binding proteins. There's a common fold in here, and the common fold is basically the palm part. So the fingers are somewhat variable, the thumb is somewhat variable, but there are a lot of these proteins out there, and this is an RNA grasp fold, and you'll find it in all these different proteins, and also in polymerases. So polymerases are an RNA grasp in the center with fingers and a thumb. They're going to help to close in the right nucleotide once it's grasped it, and usually a metal ion that will help to sort of cement that, covalently bond that for you chemists, yeah, onto the end of a chain. Yeah, so yeah, a polymerase is just an RNA grasp fold. Easy, no big deal. And this is showing what each of these things will do. Yeah. All right. Here's the slide I mentioned before. So we've got a family tree here of different kinds of plants and animals. And so uniconts, you remember those from biology 2 maybe? These are all your chromalveolates. These are all your single-celled organisms. Some of these things can move around and look at you with primitive eyeballs and still be single-celled and do photosynthesis and swim around and, live their best lives. They're kind of all the things in between what we would consider plants and animals. And really they aren't in between. They're just around. They're the rule. We're a weird exception, I would say. And so this is showing that, okay, there are all these animals. And then it's showing that we have viruses that infect these things. And so, you'll have some groups of what they call picornaviruses. So those are things like poliovirus. And they will only infect land plants. And so they're showing different virus genera in there that infect them. And there you go. You get some other groups. Totiviruses seem to do very well in excavates only, and so on. Uniconts have their own set of viruses, and chromalveolates have their own set of viruses. So this would be supported support for, well, the paper is called The Big Bang of Picorna-like viruses. So the idea that life and viruses have basically been evolving together for a long time or all of time. But it's saying that Picorna viruses, it says that they must have come before the eukaryotic groups split off from each other. So around the time of eukaryote number 1, the last universal common ancestor. And that's a bummer, because you're the last common ancestor of literally everything on Earth, and even you have to deal with viruses? I mean, where does it end? Oh my goodness. Yeah. All right. Shown here, these are some of the virus groups, and they're mapped on here. And so you can see that virus groups that are infecting similar organisms are generally going to be the same color, which means they are from that colored group that was up here. So that's just showing that there's some congruence between the virus phylogenetic tree and the eukaryote phylogenetic trees. And also you can see enough colors mixed around together that you might say, well, all right, that may be, but there are also a lot of exceptions. And I think as time has gone on and people have found more viruses, there are a lot of exceptions. So it's not necessarily a bad idea, but just an idea that is very difficult to support from the available data. And without a time machine, I don't know that you can ever actually do that. So, all right. There's a guy called Eugene Koonin who has made a living studying the origin of viruses, early evolution of viruses, radiation of viruses. All of his papers are really good, and he must be really old these days, but I'm glad he's still around and still producing things. And if you read this in the future and he's gone, left a heck of a legacy, that's for sure. So this is from one of his papers, and he's talking about, like, okay, where did some of these RNA viruses come from, these Picorna-like viruses? And so he's looking at what genes they have. And if you did the extra credit project, and if this year's extra credit project was virus annotation, then you will have looked at some of the genes that are common to different kinds of viruses. And that's pretty much exactly what he's doing. And so he's trying to say, all right, what's common? So we usually have a polymerase. It usually has a protease right before it, except for a couple of weird cases. And so probably the ancestor had, this right before. Usually when there's a VPG, remember that's the little protein that works instead of a cap on picornavirus RNA and some other kinds of virus RNA. And I don't know if DNA, but at least RNA. Then that's usually right before the protease. And sure enough, there it shows up right there. And so it's like, that's probably the thing. And quite often the last protein in there is going to be the polymerase. And or rather the capsid is your JRC, the polymerase is blue. And so that you will find actually moving all over the place, but it's a little more common in last position than in first position or on its own. And so, sure, why not? Maybe. Could be. Jelly roll capsid. And so it's more a think piece to get you appreciating this particular world of viruses than a thing that definitely happened, but definitely could have happened. And kind of interesting to think about, because if you understand stuff like this, if you understand that all of these picornaviruses, if you look what they have in common, well, most of them have a 3C protease. So if you wanted to go in with an inhibitor that would knock out multiple picornaviruses, you might be thinking 3C protease. And that's not a bad thought. But you'll find some of these that don't, like Partiti viruses. And so if humans were infected by Partiti viruses, which I don't believe we are, but again, I don't know, check ViralZone, and people discover new things all the time. Those would be a bad ones that you would miss with that approach. But looking at these, the one thing that is always, always, always in common is not even the superfamily 3 helicase, the S3H, which is sometimes replaced by an superfamily 2 helicase, but it's the polymerase. And so maybe polymerase inhibitors are the thing to go for. And if you're thinking polymerase inhibitors, yeah, which part should I inhibit? Well, maybe inhibit the center of the polymerase, the part that is the RNA grasp fold, the ancient machine that, even though viruses can change everything all the time, this is a part that they are still using, because basically when they change this part, they're dead. Yeah. So that's not a bad thing to go for. So that's the sort of understanding we're looking for here. Now in the second part, we'll go into some of these theories and look at a little bit more of the data, but this is enough for part one. You got to keep it bite-sized. All right, see you in a minute.